Namaste everyone, welcome back to the new video. Today we will learn ELISA or ELISA test. Enzyme linked immunosorbent assay is also known as enzyme immunoassay or EIA. ELISA basically it is a biochemical technique to detect and quantify the presence of specific proteins. Which are those proteins? Antigens, antibody or hormones. So ELISA were first developed in the early 70s as a replacement for radioimmunoassay or RIA or RIA. ELISA is based on the principle of antigen-antibody interaction. The antibody is a protein produced by organism's immune system and an antigen is a protein coming from foreign source. So basically ELISA is made for the detection of proteins, and antigen is also protein and antibody is also protein. So we can detect antigen, the amount of antigen or antibody by using ELISA test. So how ELISA test perform, or what is ELISA test? The amount of compound, that means the compound we need to detect, that means the specific protein, the amount of compound corresponds to the substrate converted to colored product by the enzyme linked antibody, and by using colorimeter we detect the intensity of color and the amount of protein in the biological sample. You will understand whenever we see the process. We will learn the process in the next part of the chapter. Next example where we can use ELISA test. ELISA successfully diagnosed HIV infection, Zika virus, rotavirus, syphilis, and we can use ELISA test in pregnancy tests. Next, end results. What are the end results of the ELISA test? The more the intensity of the color change, the more the concentration of protein. When it comes to disease infection, it can be detected by the presence of antigens, which are ultimately proteins, and ELISA test is made for the detection of amount of proteins in biological samples. Next, requirements. What we require to perform ELISA test? First, microtiter plate. We need microtiter plate consisting of multiple wells, and each well should be coated with polystyrene because polystyrene facilitates the adsorption of compound to the well. Next, we require compound to be detected. It may be antigen, antibody or hormones. Next, we require primary antibody which is specific to compound which we are going to detect. Next, we require enzyme linked secondary antibody which is specific to primary antibody. Next, we require colorimeter or ELISA plate recorder to check the color change as substrate binds to the enzyme. Next, we require colorless substrate. Next, most common enzymes used in ELISA are first HRP, that is horseradish peroxidase. Next, AP, that is alkaline phosphatase. These enzymes converts the substrate into colored product. Let's take example of substrate. Substrate TMB, that means 3355 tetramethylbenzidine. It produces orange color after reacting with the enzyme HRP. Second example, substrate OPD, that means orthophenylene diamine, produces blue color with the enzyme HRP. These are the examples of substrate. It's fine. Let's study types of ELISA. Three most common types of ELISA are direct ELISA, indirect ELISA, and sandwich ELISA. In the direct ELISA, compound is attached to the polystyrene plate. Then the enzyme linked secondary antibody is added. and which is bind to the compound, and then the wash is given by using PBS, which is phosphate buffer saline. After that, substrate is added, which is then converted to colored product by using enzyme linked secondary antibody, and this colored product is measured by colorimeter. This is all about direct ELISA. Indirect ELISA. In the indirect ELISA, compound is added to the polystyrene plate. Primary antibody is added which binds to the compound. Then wash is given and the enzyme linked secondary antibody is added. Again the wash is given and the substrate is added, which converts into the colored product and measured by calorimeter. This is the indirect ELISA.